Hello and welcome to Effortless Mastery. Today, we'll be continuing our series on embryology, general embryology. And today we'll be learning about spermatogenesis, a very exciting topic. Have you ever wondered how are sperm cells produced? Then you are in the right class, spermatogenesis. But please, before we rush into this video, if you've not watched our other videos on this playlist, please ensure you go through them. They will help equip you and help you understand embryology better and better. But for today, if it's your first time, please go ahead, stay on the video, you understand fully. Now, what is spermatogenesis? Spermatogenesis, if I'm using the root words, spermato and genesis, the word genesis means development. Genesis means development. So in this case, it will be development of sperm. Some people, instead of saying development, could say formation. So as a way of definition, spermatogenesis is the formation of sperm cells from what? Germ cells, the male germ cells. So that is the exact definition. So development and formation of sperm from the germ cells. Development and formation of sperm from the germ cells. So like I've cited, spermatogenesis, it in males, it occurs in their mixed male gonad, and the exact point is the seminiferous tubules of the testes. Of course, we know the male gonad is the testes. So within the seminiferous tubules of the testes, that's where spermatogenesis occurs, formation of sperm. Now, it usually lasts around 64 to 72 days. Of course, the range differs, but 64 to 72 days, and then it begins at puberty. As we go on in this series, you get to hear about development for females. You realize that development for females begin intrauterine within the mother's womb. But when I come for a male, the development of sperm is going to start at puberty. So up until puberty, nothing like that happens. So that is when it starts. Now, what else is important? So when we say development, we realize everything called a sex gamete. A sex gamete in this case for males is sperm, for females is what? Matured ovum. Some people just call it egg, and that is very correct. Now, how do I move from primordial germ cells? Please, the word primordial means first of its kind. How do I move from primordial germ cells here? Who is even primordial germ cells? So it's important we know that the source of every sex gamete, whether in male or females, is gotten back from primordial germ cells. And this primordial germ cell, the word primordial means first of its kind. The word germ means you are used to form something, that you are used for procreation. So the idea of primordial germ cell is the first cell that is going to form your that is going to be used for germination, or in this case, fertilization. So when we look at it very clearly, the primordial germ cells originate from the surface of the epiblast during week 2 of intrauterine development. So during week 2, primordial germ cells will develop, and from that week 2, these germ cells now arise from here and then get to the wall of the yolk sac, and from week 4 they move from the wall of the yolk sac and they arrive at the gonads at week 5. So once they arrive at the gonads, so let's think of it. If the fetus, in this case, of course, let me just say the child, while it was in the mother's womb, if the child was a male, of course, they were arriving at the testes. And what happens? Once primordial germ cell gets to the testes, he differentiates and becomes spermatogonium. I had to use different pens so that you could understand. Spermatogonium. So the primordial germ cell, just because by virtue of getting to testes, I now call him spermatogonium. He differentiates. But if it was to ovary, I'll call him ogonium. Now, the process by which the spermatogonium changes to our matured sperm is what we call spermatogenesis. Whereas the process by which ogonium changes to the matured ovum is what we call oogenesis. So for today's class, we'll be focused on spermatogenesis. All right, so like earlier stated, spermatogenesis occurs in the seminiferous tubules of the testes. This is the duration and it begins at puberty. Now, the lovely thing about the stages of spermatogenesis, moving from spermatogonium up until the sperm, is actually that it contains 3 major stages. We have mitosis, we have growth. Okay, maybe I would say this is a transition. We have meiosis, both first and second, and then we have the last guy called spermiogenesis. So you realize that everything begins as spermatogonium. Now, the spermatogonium in the testes, within the testes, at puberty, will now start undergoing mitosis. Now, what we are told is that at first, these guys are we differentiate spermatogonium into two, type A and type B. Type A is majorly for self-renewal. So I just divide to ensure we are still there. I keep multiplying. So like every single time we do mitosis, you divide to give two of your kind. Divide to give two of your kind. But when I come to type B, the sense of changing to type B so that type B can go ahead to enter. Type B can go ahead to enter the spermatogenic pathway. Thus, it can continue down to become primary spermatocyte. So the key role of this other one was self-renewal, whereas the key role of type B is to actually initiate or continue spermatogenesis. All right, so during the first stage, spermatogonium, specifically type A, undergoes multiple mitotic divisions. And as it undergoes mitotic divisions, please, there is no complete cytokinesis. So the cells are still kind of ahead to each other. They're still joined to each other, but they do the normal cell division. So I'll keep dividing, forming many of type A, but gradually one of the type A will now change to type B, which is type B spermatogonium. But the key thing to see here is that the diploid number is maintained. Why? It is still mitosis. After this, there is growth in size from spermatogonium to primary spermatocyte. So the key difference between spermatogonium and primary spermatocyte is just a growth in size. And then, but it maintains the diploid number. So after the growth, I come to meiosis. But when we do the first meiotic division, as expected from our videos, when we learned about meiosis, you realize there will be halving of the chromosome number from diploid to haploid. So we have secondary spermatocytes. And from here, the secondary spermatocytes does second meiotic division. And once I do the second meiotic division, what do I have? Spermatids. But then this is not where it stops. The future sperm, or would I say the final structure of a sperm, expects that the sperm should have a head containing a nucleus. So it has a head, a middle piece, and a tail. The head contains nucleus and acrosome. I'm talking about the most important things. The middle piece will contain a lot of mitochondria for energy, whereas the other side is the tail, which is flagellum. Now, so how do I move from a normal cell, which is round? You know how a normal animal cell should be? Round, having a nucleus in the center, having other organelles. How do I move from a normal cell to this classical structure or sperm or spermatozoa? Now, this process of changing from here to here is what we call spermiogenesis. So spermiogenesis is just the differentiation of spermatids to spermatozoa. Differentiation of spermatids to spermatozoa. So now you observe, what are the key things that changes? Number one, amongst others, number one is that there is Golgi apparatus will change to acrosomes. Golgi apparatus will change to acrosomes. So they will just come together. And when they come together, what they fuse to form is acrosome. Acrosome is important for a reaction we call acrosomal reaction. It is usually done during fertilization, during one of the phases of fertilization. One of the things we'll learn in the course of this playlist. Now, beyond that, the centroles, of course, are people that are cytoskeleton important in the cell division. Those centroles rearrange to form flagellum. Then the third thing that happens is that the cytoplasm, there's a loss of a lot of the cytoplasm, so that I just have small cytoplasm left, and then the nucleus condenses. So there are four things that happen, or would I say the process of spermiogenesis involves four things. Number one is that acrosome, Golgi apparatus come together to form acrosome. Flagellum is gotten from centroles. Cytoplasm is lost. A lot of cytoplasm is lost. Then a nucleus condenses. And this is the final picture called spermatozoa. So now, also very important is that during the course of spermatozoa development, which is spermatogenesis, you realize that there are some cells that help to support. So the nourishment you need, that the developing sperm needs, the nourishment, every other thing it needs, just general support is provided by this cell we call cetolic cells. Some people also call it substentacular cells. Other people just call them supporting cells. So these cells, their main role from the name is to support the developing sperm. That is their main role. Now, beyond that, we have another set of cells called Leydig cells in the testes. And the Leydig cells, which are actually interstitial cells, are responsible for production of the male sex hormone called testosterone. And since I've mentioned hormones, there are 4 hormones that are involved in the regulation of spermatogenesis as a whole. Those hormones include GnRH, which is gonadotropin releasing hormone produced by the hypothalamus. We also have follicle stimulating hormone FSH. We also have luteinizing hormone, both of which are called gonadotropins, and they are produced by the anterior pituitary gland. Then finally we have the testosterone, which is the male sex hormone. Like I've said, it's produced by the Leydig cells of the testes. So what is very key to note about these hormones? Their function. The idea of gonadotropin releasing hormone is that they are released. These hormones are people that will cause the release of gonadotropins from the pituitary gland. So the source here is pituitary gland, source here is hypothalamus, source here is testes. Now, gonadotropins, from their name, tropins are things that causes something to grow. So when I say gonadotropins, these are people that will cause the gonads to grow and develop. So, and then produce what they should produce. So the luteinizing hormone specifically is the one that will act on Leydig cells to cause production of testosterone. The action of follicle stimulating hormone in males is majorly on Sertolic cells to promote its support. And then the male testosterone or male sex hormone is the one that will be responsible for the characteristics we see in male following puberty, the normal regular things, the voice, and then beyond the voice, the beards, those kind of the pubic hairs and all of those things that are seen. Those male characteristics, secondary characteristics, are all attributed to testosterone. And then also it plays a role in spermatogenesis. So, and that brings us to the end. So in summary, what have we learned? Spermatogenesis, a process whereby sperm is being formed from germ cells, primordial germ cells. Where is the source of these primordial germ cells? Epiblast cells. And they develop in week 2, move to the yolk sac. From the yolk sac, they end up getting to the gonads, respective gonads. In males, that will be testes. Now, when this happens and they get to the testes in males, they become spermatogonium. Now, the process by which this spermatogonium changes to sperm is what we call spermatogenesis. Now, first it is from type A, mitosis will change it to type B. More mitosis and growth will now change one of these to primary spermatocytes, still diploid number. After the first meiotic division to form secondary, we move to haploid. After second meiotic, it is spermatid, but still haploid. Then we now have a process called spermiogenesis, where the spermatid, which were initially like every other animal cell, changes to a conformation that has head, middle piece, and tail. So the case is, what are the key changes? The Golgi apparatus become acrosome, centros become flagellum, rearranged to form flagellum, and then the cytoplasm, a lot of the cytoplasm is lost, so we say cytoplasmic reduction, and then the nucleus condenses. So the final picture is now the head of the sperm with nucleus and acrosome, the middle piece with a lot of mitochondria, and then the flagellum. Now, what are the key hormones here? Testosterone, follicle stimulating, luteinizing, and gonadotropin releasing hormone. It is important to note that spermatogenesis, which happens in males, is only specific. It only begins at puberty and it can last the entire lifetime. Thank you very much for joining us today. I believe you understand for sure. So please do well to subscribe to this YouTube channel. Do well to like this video. Do well to comment. Any form of issues you have, you can mention them in the comment section. If there's a particular video you want us to do, please mention it in the comment section. Ensure you share these videos to as many people as possible so I can help others to